Hi, my name is Eddie Polanco, and today we're going to talk about how quantitative phase imaging can be used to help pair the right cancer patient with the right cancer therapy. And so I know we all know that this is what precision medicine is all about. However, I want to start by talking about two major categories that were identified by Friedman et al. In 2016. The first is targeted precision medicine, which is all about trying to use a genetic profile of tumor cells to find known targetable mutations. This increases the likelihood that the therapy will be effective. However, there are two major drawbacks. The first is that not all patients have a targetable mutation, making it impossible for this approach to even help them at all. The second is that often targeted therapies exhibit only a partial response, failing to wipe out the entire tumor, which can often cause the tumor to grow back even more aggressively than before. This is often caused by genetic heterogeneity within the tumor, which is difficult to identify when you're doing genetic profiles, and is and it arises from the fact that not all cells in the tumor are exactly the same. The second major category is functional precision medicine, which is all about using a functional assay in order to determine whether or not anticipated therapies will have a measurable effect on tumor cells, on actual tumor cells from a patient, in order to guide their therapy. So this is what the workflow for functional precision medicine looks like, and it starts with a patient who has a tumor, and the tumor is biopsied in order to validate malignancy. Biopsied cells can be grown up in culture and then seeded in one of these various types of model systems in order to perform a functional assay. As we go down this list, these different model systems become more representative of the tumor microenvironment. However, progressively more cells are required in order to use these types of assays, so it requires a longer expansion time over here. Now, that said, even with having a longer expansion time, the assays then also take longer to do because you need to grow up the tumor in the mouse, or you need to establish your organoids after seeding them. So what we want to try to do is we want to see if it's possible to skip this step altogether and just biopsy enough cells that you can go directly to the assay. But in order to do that in this study, we're just going to focus on these 2D in vitro experiments in order to determine whether or not we can use in order to use the fewest number of cells possible, in order to shorten the expansion time as much as possible. And so there are a couple of different approaches for using these 2D models. The gold standard and most clinically relevant assay is called cell titer glow and is used by our collaborators at the Huntsman Cancer Institute at the University of Utah. So what we're going to do is we're going to take our assay and we're going to compare it to the gold standard, which what it does is it's an endpoint assay that measures ATP levels in samples that were exposed to therapies relative to the control in order to determine cell viability. And so we're going to test our assay against this gold standard approach in order to validate that it has clinical relevance. And so our approach involves using quantitative phase imaging to measure the mass of individual cells by measuring the phase shift as light passes through them. So quantitative phase imaging measures cell mass, and the way it does this is by measuring the phase shift as light passes through the cell, and so we can get a distribution of phase shifts through the cell. However, because the phase shift of light as it passes through a cell is linearly related to the mass of the cell, we can also compute a mass distribution in this cell. And then when we want to determine the mass of the whole cell, all we have to do is integrate over the cell. And so by QPI measures the change in mass over time as growth. So what we can do is we can repeatedly measure the cell's mass in order to determine how quickly it's growing. And so here you can see these are repeated mass measurements over time, and this cell demonstrates robust growth the entire experiment, as you can see, because it continues to gain mass throughout the experiment. So the technique that we use for quantitative phase imaging is called differential phase contrast. The way that this technique works is by using a standard compound microscope with both an objective lens and a tube lens, but replacing your light source with an LED array. The LED array allows you to quickly switch between illuminating one side of the array or the other in order to capture extra information with the objective that otherwise would have been lost, from which the phase can be recovered. In this example, we're going to show how it works illuminating from the bottom first and then from the top. And from this, the phase gradient can be computed in the y direction simply by subtracting the top minus the bottom, and dividing by the sum. From this phase gradient, we can repeat this process for the left and right halves of the LED array in order to compute the phase gradient in X. With the two phase gradients that are orthogonal to each other, we can then compute the phase shift through the cell from the phase gradient. So growth is measured as the change in mass over time. So you can see here, after determining the phase shift through the cell, we're able to determine the distribution of mass in the cell. So in order to compute the mass of the cell, we just have to integrate over the area. We can see here with this cell that the cell has more mass in the center, where we expect the nucleus to be, and less mass on the edges and in this protrusion, where we expect the cell to be flattening out. So in order to determine cell growth, we compute the cell growth as the specific growth rate. In order to compute that, what we do is we average the mass of cells at different time points together. And in order to be able to average them like this, we normalize their mass by the initial mass of the cell. This way, every cell starts out with a mass of 1 and will continue to gain mass throughout the experiment until it doubles in mass and eventually divides. To compute the growth rate, we fit a line to this growth curve, and then we use the slope of this line as the specific growth rate, which has units of 1 over hour, because this is the slope of a normalized mass, so this is not in picograms, it's just in arbitrary units. So here, what's great about quantitative phase, one of the important advantages of quantitative phase, in fact, is the fact that you can measure the growth at every time point because you have such high resolution in mass. And so what's really cool about this is that, for example, with this particular drug, we can tell that after being treated with the drug, the cells grow pretty robustly, similar to the control, indistinguishable from the control even, for the first 10 to 12 hours. After that, we can start to see a separation in their growth rates because the 5-fluorouracil treated cells start to grow a little bit slower than the control, and we can see that here. Eventually, after about 18 hours, the 5-fluorouracil treated cells start to die, which, seeing this, the dynamics of this response would be impossible using a time average measurement such as an endpoint assay. So the way that we do this is using an automated system where the microscope travels from well to well, imaging each condition one at a time. We can do one imaging cycle every 20 minutes. Each well contains 9 imaging locations. With 96 wells, there are 864 total imaging locations. With 10 to 20 cells per imaging location, in a single experiment, we image 10 to 20,000 cells over the course of 24 hours. So, using that same strategy we just showed, where we can average the cell's mass at each time point, fit a line to it, and then determine the specific growth rate, what we do is we compute the specific growth rate for each well in the plate. And we show it just like this. So it looks just like a 96 well plate with 96 specific growth rates. And you can see here you can readily see at a glance the specific growth rate in each well. It's pretty clear something like doxorubicin, which is highly cytotoxic, where even at middle concentrations, the cell's growth rate seems to be pretty dramatically affected. However, even at the highest concentrations, their growth rate is affected even more, even negative in some cases. However, another drug like fulvestrant, you can see that the cells grow pretty robustly at all concentrations. Even at the highest concentrations, the cells seem to grow pretty well. And when we plot the specific growth rate as a function of concentration, you can see that here. We can see a fairly resistant line, or that the cells are fairly resistant to fulvestrant in particular, because at all concentrations, the cells grow fairly robustly. However, we can also see how cytotoxic doxorubicin is and how sensitive the cells are to this drug, because the growth rate drops off fairly dramatically, fairly quickly, as you increase the concentration from the lowest to even just the next lowest or two. So we repeated this experiment with MCF7 cells in order to show that we can measure sensitivity and resistance in other cell lines. So here you can see that, again, the dots are on the left, looking at the specific growth rate plot, that these cells look very sensitive to doxorubicin. Even though they're not quite as affected in the middle concentrations, they're pretty dramatically affected at the highest concentrations. And here, when we look here, it's a little bit unclear whether or not they're as affected by, they're resistant to fulvestrant, but when we look at the plot on the right, again, it's still a little bit unclear. So maybe there's some more work we can do there in order to clarify that particular drug. However, you can see when looking at the dose response curves that there is some effect on the cells from the 5FU as well as with the hydroxytamoxifen. And so one of the things we want to do is we want to see if we can see sensitivity to these three drugs with cell titer glow to make sure that we're finding the same results. And so when we look at the cell titer glow data, we can see that we do see strong sensitivity to doxorubicin, and especially to hydroxytamoxifen at the highest concentration, which is what we're seeing in the QPI experiments as well. 5FU, you can also see a pretty strong response to it. It's actually a fairly similar response as we see with the QPI data. Now, the curve fitting for these two experiments is a little bit different, which results in slightly different IC50s, especially with this 5-fluorouracil treatment group. However, it looks like over here, maybe we didn't find exactly the extent of response that cells will have with 5FU. So we might get a better data out of this if we go to slightly higher concentrations, and maybe our curve fitting approach can be made better as well, or can be improved as well. And so then we did the MDA MB231 cells again with a second drug panel. And here we can see that they're more sensitive to some of these drugs, like the burnapant and the carboplatin, and especially to this vinblastine. And we can see a very similar response in the QPI experiments as well. One thing that I would like to point out is how there's this weird effect from docetaxel that we can see in the cell titer glow experiments, where the cells seem to be pretty dramatically affected even at the lowest concentrations, and we're measuring that with the QPI experiments as well. And so while the curve fitting is not exactly the same, and maybe the experiments can be improved just a little bit in order to get better correlation between the measured IC50s, qualitatively, the dose response curves look very, very, very remarkably similar. And so one of the important advantages of QPI is that QPI resolves growth rate at all times, which is really important for planning experiments. For example, if we just did a time average measurement at the end, then we would never know that it would be impossible to know that 5FU doesn't start to affect the cells until almost halfway through the experiment. So this might result in 5FU having a slightly lower depth of response that we wouldn't have otherwise expected without looking at data like this. So that could be another way to try to fix something like this, where the 5FU had such a dramatically different response in QPI as far as the measured IC50. And so when we look at the bottom plot, we can see here, though, with this one that was measured to be extremely cytotoxic, doxorubicin, now we can see why. It's because it has a very dramatic effect very early on, and so for the entire experiment, cells are almost dying rather than just growing robustly with the control cells before slowly dropping off. Another really important aspect of this is the ability to see just the difference in growth rate, and the fact that the 5FU does have an effect here. Even though it's not killing the cells, it is, and their cells are still growing, they're not growing as fast as the control, which is another interesting feature of this data. And so one other really important aspect of quantitative phase is that we can get heterogeneity from the single cell data, because here, for example, we're binning how many cells or what fraction of cells from the whole population have each growth rate based on their drug treatment. And so you can see, even with just the DMSO treated cells, the control cells, all cells don't grow exactly the same. We have some slow growing cells, some fast growing cells, but most cells grow somewhere in between. When we look at something like doxorubicin, we can see that the distribution is biased very much towards the very low growth rates, even negative growth rates. But then when we look at something like fulvestrant and lapatinib, something that was demonstrating resistance in one of the lines we looked at, you can see that the growth rates are pretty widely distributed. So it's good to be able to, this is something where we would be able to help us identify something like a partial response. And we can identify whether or not it's just because it's only affecting the cells a little bit, like it's causing all the cells just to have a slightly reduced growth rate, like we were seeing with the 5FU, or it could be because some cells just aren't responding and some cells maybe just have the same growth rate as the control. And so that's something that needs to be investigated further. But the single cell data and the time resolved data both together will help us be able to get at that type of question. And so in summary, quantitative phase imaging measures the growth rate of individual cells by measuring their change in mass over time. And we just talked about why that's so important, because that in combination with these the measurements of heterogeneity with the single cell data, this is something that allows us to start looking at questions of partial responses and identifying partial responses and trying to determine what causes them. Future work is going to continue to demonstrate the clinical relevance of this tool by demonstrating that it works comparably well to cell titer glow. And then we also plan to use data from patient-derived samples in order to compare against patient outcomes in order to determine that this tool has predictive capacity. And so with that, I would like to thank the Department of Defense for funding this work. And I would also like to thank our collaborators up at the Huntsman Cancer Institute at the University of Utah, Phil Bernard and Brian Whelm. And so with that, I will take any questions. Thank you very much.